My name is Carl Whitwer. I'm from the University of Utah, and I'm pleased to present to you today work on high-resolution DNA melting analysis, also known as high-resolution melting, sometimes abbreviated as HRM. I'll also introduce a variant of high-resolution melting called high-speed melting, or HSM. You should be looking at a mountain view surrounding Salt Lake City, Utah, which is my institution and city. And you're looking at the University of Utah below the mountains. So melting analysis has a short history. In terms of looking at melting analysis in the context of PCR, melting analysis really was combined with real-time PCR in about 1997. And it was done so both looking at probes, the melting of probes, and also the melting of dyes. We'll spend most of the time talking about high-resolution melting, looking at melting analysis in a high-resolution mode. And that really began around 2003 or so. It's been very popular for genotyping methods, including small amplicon melting, unlabeled probe melting, and snapback primer melting. We'll also talk briefly about scanning, high-resolution melting for scanning for variants, particularly heterozygotes. And we'll also talk about copy number analysis. And then near the end, I will introduce high-speed melting, HSM. So to begin our journey, modern melting analysis is performed after PCR. It's very convenient. With real-time PCR, as you know, as you advance the cycle numbers, at some cycle you'll begin to see fluorescence. That fluorescence can be from dyes or it can be from probes. And that increase in fluorescence can be used to detect a product, and the spacing, the cycle of quantification, CQ, also called CT or CP, all refer to fractional cycle numbers that allow quantification, major application of real-time PCR. Now, you can think about melting analysis as the opposite or conclusion of real-time PCR. If real-time PCR increases fluorescence, melting analysis actually melts the final product and their fluorescence decreases. So the red line is the actual raw fluorescence. The dotted line in the middle is often how the data is presented as a melting peak. It's a derivative plot because it's easier for us to identify peaks often than it is decreases in absolute fluorescence. So the main advances that have gone into melting analysis becoming popular is that with real-time PCR, we're using fluorescence instead of absorbance. You can use dyes or probes, but most of high-resolution melting is focused on using dyes, inexpensive dyes. The other real advance is that melting analysis can be fast. So it used to be when absorbance was used for DNA melting analysis that it would take 4 to 6 hours, often run overnight, to get a single melting curve. And you'll see that high-resolution melting can be done much faster, on the order of minutes. Now, if you think about real-time PCR, there's really not much to it. There's only really 3 parameters. You're keeping track of time, you're keeping track of temperature, and you're monitoring fluorescence. So those 3 parameters, time, temperature, and fluorescence, give you the information from real-time PCR. And if there's an overall increase in fluorescence, typically that allows you to detect a particular specific product. If you monitor fluorescence just once each cycle, that's typically used for quantification. And if you look continuously at fluorescence over temperature with changing temperature, you have an analytical technique. Now, if we look at those 3 parameters, time, temperature, and fluorescence, and we plot them 2 at a time on a regular graph, there are 3 different combinations of those, right? We've got time versus temperature, or time and temperature. We've got time and fluorescence. And finally, we'll look at temperature and fluorescence. So first, let's look at temperature and time. We'll look at two different fluorescence methods, dye, cybergreen 1, on your left, and then hybridization probes on your right. If you plot temperature versus time, you follow just the heating of your sample. The temperature traces I'm showing here are part of what's typical for rapid cycle PCR. So it's not long cycles. In fact, if you look at the time, it's actually listed in minutes, and typically in rapid cycle PCR, you're done in 15 to 30 minutes. Okay, so this is the first picture looking at just temperature versus time. Let's leave time on the x-axis, the horizontal axis, and instead of temperature, let's look at fluorescence. And this is the real-time part, the quantification part of real-time PCR, because the fluorescence each cycle you'll see increases, whether you're using cybergreen 1 or hybridization probes. At least it increases at certain temperatures. Now, at very hot temperatures, the double-stranded products denature or melt, and the fluorescence goes down to 0. And similarly, you can melt the probes off so that during each cycle, the fluorescence basically goes down to 0. But if you look at the top of the curves and just take fluorescence once each cycle, then you have the real-time plots. Now, this becomes even more interesting if we look at the last combination of two parameters. So we won't look at time anymore, but we'll actually look at fluorescence and temperature. So we're putting temperature on the horizontal axis and now looking at fluorescence, cybergreen 1 and hybridization probes. Here's cybergreen 1 continuously monitored during PCR. You get these curious spirals from denaturation to annealing and then an intermediate extension. The most interesting part of these curves is as you're heating up to denaturation, your product actually melts. And you can get a melting temperature for those products, or a product TM, of what you've amplified. Similarly, with hybridization probes, you get curves, and that rapid decrease in fluorescence is the probe TM, or probe melting temperatures. So melting analysis really came out of just continuously looking at fluorescence and plotting it against temperature. This was originally performed, the first melting analysis with real-time PCR was done on the light cycle or light cycle prototype, is what I'm showing right now. And here's the actual data we got off of that particular prototype for melting curves of different products. We were using cybergreen 1, and different products, depending on what was amplified, appear as different curves, appear as different melting curves. So you can use melting analysis to define or categorize PCR products by melting temperature, TM. Now, it's similar to a gel, except a gel will separate by size, and melting analysis will separate by melting temperature, TM, or the curve shape identifying specific PCR products. And this sort of led to what we ended up calling the light cycler paradigm. We were using rapid cycle PCR, so PCR is done in 15 to 20 minutes. Those little red dots are the actual acquisition. If you acquire fluorescence once each cycle, then you have means of detecting or quantifying PCR, as we discussed. In addition to amplification, melting analysis typically today is done at the end of amplification, an analytical technique that you tag on to the end of PCR, and that gives you product identification, or if you're using probes, it can give you a genotype. So there's two different components, amplification and melting analysis is what's mostly become popular today. When you use probes, instead of old-time incubation in dot plots, I like to think about melting analysis as a dynamic dot plot over temperature. So in this case, we'll use a couple of different probes, adjacent hybridization probes that are both labeled. And if you slowly increase the temperature, the stability of a probe matched to your target will be greater than one that is mismatched to the target. So if you end up plotting fluorescence versus temperature, you'll have, in the case of a heterozygote, where both the match and mismatch are present, you'll get two separate decreases in fluorescence as each allele melts from a specific probe. And you can plot that either as fluorescence, or as we discussed before, you can plot that as a derivative plot to see the melting peaks, identifying in this case the two heterozygous alleles of this particular variant. So dynamic dot plots for allele analysis. A very typical example, dual hybridization probes for factor V Leiden. The derivative plots, heterozygotes showing two peaks, the homozygotes, depending on what they are, showing the other two peaks in black and blue. This kind of testing was actually the first genetic tests that were approved by the FDA in the US. That was around 2003. And factor V Leiden and prothrombin were the first genetic tests to be FDA approved using these hybridization probes and melting analysis. So in terms of products and monitoring product amplification, people ended up using CyberGreen 1 almost as a quality control of what you were amplifying. The expectation was that if you were amplifying a single product, you would get a single peak. And then you would feel confident that you weren't amplifying multiple products and that you probably had what you wanted. So this is what you expect, a nice single peak. Now, with that mindset, if you were to see a melting curve that had multiple peaks in it, you might think that this is bad PCR, poor PCR that was amplifying multiple different genomic products in this case, or primer timers. But it turns out not to be true, and this really turned us on to the potential power of melting analysis. So what you're seeing here, even though it has three peaks, is a single PCR product that's melting in multiple domains because the distribution of GC and AT base pairs is not equal throughout the amplicon. So we decided to focus much more on the melting analysis and tried to build better and better melting instruments. And that was the origin of high-resolution melting. Here's some of the initial prototypes that we worked with. We were working with Roche at the time. And the instrument on the left we call the Volkschmelzer. It turns out that if you're German, you don't use that term. We thought we were being cute, but it turns out that that was the wrong name for the instrument. Eventually we renamed it, and there's a couple of proud parents in your lower right for the first prototype and actual commercial version of high-resolution melting. People were interested, so here's almost an exponential growth of high-resolution melting in the published literature from 2003 up to about 2011. There was something there that people liked. They liked the simplicity because you could look at high-resolution melting on the same instrument that you were doing real-time PCR in. At one point in a review, this is a quote, high-resolution melting was described as a multi-purpose technology and a standard tool that should be present in any laboratory studying nucleic acids. Well, PCR and everything else doesn't grow exponentially forever. So here's a log plot showing near exponential growth up to about 2011 and then a slower growth since then. It's settling down at about 800 or 900 publications per year. But certainly has made itself known as a useful tool over time. Now, you'll end up seeing that there are many applications of high-resolution melting, but typically when someone refers to high-resolution melting, they're referring to genotyping. And usually that will be done with a small amplicon. So if you're after a sequence variant, you'll place your primers on each side of the variant, and by melting analysis alone, no probes, no fluorescence probes, just a dye, you can identify the different variants. In this case, in SNP, there are 3 curves. The homozygous curves, one in black on the right and one just shifted in dark gray, they're almost parallel, but they are different in TM enough that if you have the capacity of high-resolution analysis, you'll be able to distinguish them. Now, the heterozygote is a different character. It actually has a different shape. It doesn't follow the pattern of the homozygotes because there's more going on than just a shift in TM from GC base pairs. So we're going to look at both of those. We'll first consider the homozygous differences, and then we'll look at what's causing that odd shape on the heterozygote. So for the homozygous differences, you've got a change in TM, a change in the melting temperature. And that's why high-resolution melting is required. You've got to tell the difference of that shift, which sometimes can be very, very small. For example, if you look at all human SNPs and plot how large that change is, how large that delta TM change is, most SNP homozygotes will differ by about a degree centigrade. But you'll see that there's variation sometimes all the way down to a predicted variation by nearest neighbor analysis of 0. That is, in rare cases, about 4% of human SNPs, there's no predicted difference between homozygotes. So that means we'll have to come up with something else to pick up that 4%, to distinguish that 4%. So it works very well on 90+% of SNPs. For homozygous differences, now the heterozygotes, that change in shape is always easy to distinguish. Now, why is that? Well, if you think about amplifying a heterozygote, you'll produce the 2 homoduplexes that you began with. So let's begin in this case just a simple SNP, a TA base pair going to a CG base pair. Those faithful homoduplexes will melt. They'll melt very close to each other, but they have the same pattern. Now, you'll also be amplifying heteroduplexes because there's nothing that says the T strand has to go with the A strand. You can form the mismatched duplexes, the heteroduplexes, and those have significantly lower melting temperatures because they've got the mismatches. Now, all 4 of those duplexes are present in the same tube, so you don't see them individually. What you end up seeing is a combination of both of those, and the linear combination of both ends up as this skewed or warped melting curve, signifying the presence of a heterozygote. So with small amplicon melting, heterozygotes are really easy to distinguish, and the homozygous differences are usually easy on high-resolution mode of instruments, but occasionally can be quite difficult to see. We've come up with some software to design small amplicon melting, particularly for single nucleotide changes, where all you really have to do on the upper left of this free web application is to enter in the RS number. And with that, you'll end up having plotted below suggestions and predictions of what the melting curves actually should look like. I'll present a couple of, or just show you a couple of software tools that are present on the site dnautah.edu as we go along. So design of small amplicon melting. This can become complex if you want to. The farthest we've taken it is to amplify 4 different SNPs at one time. These are 4 SNPs for coagulation, factor V, 2 MTHFR targets, and prothrombin. The curves start looking complicated, but if you look at each one individually, the wild type homozygous variant and heterozygous variant are all detectable and distinguishable from each other, all from just single amplicon melting. One dye, single color. As things get larger, we saw a case where you form domains, and this is a case of multiple domain melting. The variant tends to be, in this case, in the lower temperature, higher fluorescence domain. And again, when you look carefully using high resolution melting, you can distinguish the different genotypes in this particular SMP in a 500 base pair product. When you have these longer amplicons and you are concerned that they may melt in multiple domains, it's very useful to be able to predict whether they will or not melt in single domains or in multiple domains. So we have this application called UMelt, where you plug in your sequence, copy your sequence, and it will predict what your melting curves and what your domains should look like. Anyone doing PCR with dyes should really use something like this. It just makes sense to know where your product denatures and what it looks like on melting analysis. So just to review small amplicon genotyping, it really is the simplest of all genotyping methods. We're only adding a dye to PCR. We're not doing anything to primers or probes. You just got your two regular primers surrounding the area of sequence variation. It does have some limitations, as we discussed. For instance, about 4% of SNPs can't be distinguished from each other. And there are insertions and deletions that cannot be distinguished. We also have solutions, which we'll go through. So if you have really low delta TMs, low differentiation between homozygous and wild type, which you can predict, there are, I've listed 4 different methods to distinguish the wild type from variant homozygotes. The ones we'll spend time talking about are the last two, the unlabeled probes and snapback primers, because they'll provide the specificity of a probe, but just using a dye. When you look in general with melting with double-stranded DNA dyes, the idea is that you don't have any covalent labels to any fluorophores, so no expensive probes. And you multiplex by temperature, as you saw in the coagulation example of the 4 SNPs, not by color, as is commonly done in real-time PCR. So again, the small amplicon melting is the simplest way to do this, where your primers are on each side of the region you're interrogating. So the SNP or other variant would be in the red area inside the primers here. We'll also talk a little bit about scanning for heterozygotes that can be done in larger amplicons. A different way to look for sequence variants. It's also very, very simple. But for now, we're going to introduce a variant of small amplicon melting that can even distinguish that small 4% that you can't do directly by small amplicon melting. And that'll be using an unlabeled probe. So there's a probe here, but it's not covalently labeled to any fluorophore. Typically, to see the signal, you'll run asymmetric PCR, and you'll also block the 3' end of your oligo with phosphate or some other blocker so that it does not serve as a primer. So now the region of sequence interrogation is the blue and red line, which is the unlabeled, the portion of the amplicon that the unlabeled probe covers. So you can focus on a specific area within an amplicon now with an unlabeled probe. Here's the results you get with an unlabeled probe. Again, a schematic up on top just under the title. And you get 2 different areas of melting analysis. On the left, you actually have the area where the probe melts, and that looks very, very similar, if you remember, to the labeled probe example that we started off with. Now, not only do you have the area of probe melting, but because you've amplified, albeit asymmetrically, you've got the probe annealing to the product, but you've also got the product strands annealing. So that's what's going on over on the right-hand side with the amplicon melting. And those curves, the homozygotes overlap, but you'll see a double peak over on the right that actually represents the heterozygote portion. Now, if we just focused on the amplicon melting, we'd be veering into what we'll end up calling scanning. But in this particular case, there's actually two different areas that self-verify. You've got the probe melting and also amplicon melting, both indicating, in this case, the particular genotype that you're identifying with unlabeled probes. We did this on a 384-well plate, so here's factor V Leiden, and it's pretty obvious the clustering is not very difficult. The blue line, the rare homozygotes, is parallel on the right-hand side to the wild type in black. And the red line, both in the probe area, the unlabeled probe melting area, and the amplicon area, two different areas you can look at that tell you the same thing. So it's a double verification of what you've amplified with unlabeled probes. So again, here's a schematic of the unlabeled probe, and we're going to look at a variant of that that has some advantages. We're going to take that unlabeled probe and we're going to place it on the tail of one of the primers, so that tail snaps back over the region of variation. It's an unlabeled probe that's hooked on to one primer. It has a priming region, and it also has a probe element. That was originally as a 5' tail of the primer, but you'll get this intramolecular hybridization visualized with the dye in green, very similar to an unlabeled probe. And you get curves that look very similar to what we just looked at. So over on the left-hand side, you'll have the probe melting, in this case with a snapback primer. And on the right-hand side, you have the amplicon melting. Here is one complex example of using a snapback primer to genotype, in this case, a AT repeat that's responsible for Gilbert's syndrome. So the tail, the snapback primer, interrogates that region of the AT repeat. And depending on the length of the repeat, the melting analysis gives you different curves when you have instrumentation capability to perform high-resolution melting. So this was an example of a snapback primer. And now we're going to contrast genotyping versus scanning. Genotyping that we've been talking about always analyzes one locus for different variants, whereas scanning determines whether any variants are present in a PCR product. So different methods, different focus. Let's look at scanning. You can use amplicon melting to identify different variants that are within the primers. I like this as it spreads out the variation across a two-dimensional plot when it's plotted as the fluorescence difference from wild type. So all of the black curves down at 0 are the wild type curves, and we're subtracting the variation, any variation from that wild type curve, and then plotting it. So you'll see different variants show up. The blue actually is a homozygous variant of a common polymorphism. The heterozygote of that same polymorphism is shown in red. And then you have some other variants that are also present at some frequency in this particular population. The green and then some compound heterozygotes, purple and orange. So it's a very quick way to establish sequence variation and often the clustering or identity of those variants in a population. So just quickly to summarize the utility of scanning, it is inexpensive and rapid. It picks up essentially all variants better than better than other scanning techniques. Like any PCR technique, it doesn't detect large insertions or deletions. It can't detect what you don't amplify. And benign polymorphisms may require further analysis. Of course, any variant will be picked up, and you have to distinguish whether it's a significant mutation or whether it's essentially a benign genotype. And scanning has in some way been trumped by, I probably shouldn't use that word anymore, by massively parallel sequencing. The massively parallel sequencing is extraordinarily popular today because of the extensive amount of sequencing, the massive amount of sequence you can get. So for multiple gene targets, exomes, and genomes, you really have, you don't want to use high resolution melting scanning. It's a different issue if you want to do something quickly. So if you have a single gene, for instance, and you don't want to take the time required for massively parallel sequencing or even Sanger sequencing, that often has turnaround times from commercial labs of a couple of weeks. What can you do? So we're going to look at a number of different examples of the applications of high-resolution melting now. And we're going to start with scanning and rapid genetic analysis. In this particular case, the disorder is chronic granulomatous disease. And most cases are caused by an X-linked gene, CYBB. And we'll look at the high-resolution amplicon melting by scanning 13 exons. To make things easier, we'll tail the primers with common sequences for sequencing the rare times that we will detect a variant. And we also end up plating the primers on plates so we can run everything on a 96-well plate, all 13 exons. And in the end, we do sequence rare variants when they occur. So here's what the melting analysis looks like after you normalize all of the different plots. They're melting from around 85 degrees C up to about 91 degrees C. And there are actually 13 curves there. You'll notice that some are multiple domains, other are single domains, as you might expect. The base pair size here is probably about 100 to about 350 or so. And when you look at them one by one, or you can write automated protocols to look at the exons one by one, you'll be able to identify which one of those 13 exons, if any, have a variant. Now, in this case, there was a variant, and it was present in exon 7. And what you see is this change in shape that we talked about in terms of finding a heterozygote. The sample was mixed with wild-type DNA, so we picked up the single variant copy in this X-linked disorder. So the nice thing about this is you can do it rapidly. We've only populated a plate full of primers, amplified and melted them. So ideally what you can do in terms of a workflow is by extraction methods that take maybe an hour or so. The amplification also taking perhaps an hour. The scanning takes about 5 minutes. In most cases, instead of the long turnaround times that are typical, when there's no variants, no rare variants, these can be reported out within 3 hours. And most exons, at least in this disorder, and most disorders, are wild type. So many things can be reported out as negative. Now, if you run into a positive, and of course that's what we're looking for, then you can take that specific exon and end up sequencing it. But even that process, if you're focused on turnaround time, really can be done within 6 hours. So an example of rapid genetic analysis of a specific gene. Continuing with examples of high-resolution melting, this is a platform, 1-hour sample-to-answer system that's popular these days for syndromic illnesses. It's a BioFire film array. And there are 4 FDA-approved panels, upper respiratory, gastrointestinal, blood culture, and meningitis, in this relatively small instrument where all of the reagents are are pre-planned in a particular pouch. So you just add the sample, and in 1 hour you actually identify the specific cause of a syndrome. Popular, people like this. Now, it turns out that this system uses melting analysis, not real-time PCR. And we do that's done for a particular reason. So when you monitor fluorescence over cycles for quantitative PCR, sometimes at low copy numbers it's very difficult to tell whether there's anything there or not. So in this particular case, when we're looking for viruses or bacteria, this line looks pretty flat unless you look at the cycles near the end and amplify the horizontal axis, the vertical axis some, and out there around cycle 28 or 30, is that a positive, or would you call it a negative? Well, if you do melting analysis at cycle 30, you actually get a very distinct drop in fluorescence, whose derivative also looks very distinct. So this is evidence that melting analysis is more sensitive than just once a cycle fluorescence acquisition of quantitative PCR. If you think about it, that's not that surprising. You're looking at temperature changes, you're looking at fluorescence changes over temperature as the temperature changes. So another example of high resolution melting. Here's one out of New Zealand. New Zealand's unique because a lot of their scientific work is focused on not necessarily humans, but either plants or animals. And in this case, there were dinucleotide repeats that identified different types of grapevines that go into wine. So this is a quality control measure to make sure that what they were growing and processing was actually the particular kind of grape and wine that they were claiming. And they're differentiated, again, by their melting curves. Because of differences in the dinucleotide repeats, they melt differently. Possible by high-resolution melting. Here's another somewhat more recent use of high-resolution melting to establish copy number. In this case, I'm showing trisomy 21 and trisomy 18 as rather obvious examples where when you normalize a reference peak over on the left, your trisomic samples will show greater peak heights than the disomic samples. Now, the trick on doing this is instead of trying to catch a particular cycle where the quantification retains the original copy number, we use limited DPs. We restrict the DNTPs so that before the primers end up plateauing and getting rid of this quantitative difference, the DNTPs limit the amplification, giving these kind of results by melting. Here's another example for spinal muscular atrophy. This melting's probably the easiest and best technique to look at this particular disorder. It's due to different copy number changes in two genes, SMN1 and SMN2, shown on the left and the right. And the reference gene in this case is over on the right-hand side of both SMN1 and SMN2. And the copy numbers, the blue in each example is basically 0 copies of the particular gene. Red is 1 copy, black is 2 copies. And further up, verified by digital PCR, are higher numbers of copies for these two genes important in spinal muscular atrophy. Just a few more examples on uses of high-resolution melting. In this case, sequence identity. I'll just show you a quick example of using identical melting curves to identify identical sequences. So if your sequence is the same, your melting curve should be the same. Let's look at one example in terms of transplant compatibility, particularly HLA. We're going to be amplifying HLA, that highly variable region that determines compatibility. And we're going to correlate the melting curves directly to a phenotype or a transplantation result now. So in this case, we actually don't care what the genotype is. We're just going to look at, in the case of transplant matching within a family, whether the sequence is the same by looking at whether the melting curves are the same. So in this case, I'm presenting from Utah. Here's a family with 11 siblings for demonstration purposes. And all of the siblings here are going to be melted over the HLA region. And we'll look at 4 different regions of HLA-B and HLA-C, of an exon 2 and exon 3. These are the highly variable regions of these 2 loci. And what you'll notice is those 11 children in each case cluster into 4 different melting curves because there's only 4 possible genotypes or combinations from the 2 parents. Crossing over is rare in HLA. So what does this mean? Well, it means that you are transplant compatible with everyone in your particular cluster. So if you're in a cluster with a lot of siblings, that means there's quite a few choices. There is one case here, one sibling number 8, that had no particular match. So a method of very rapidly, without genotyping, determining sequence compatibility just by melting curve identity. The very last thing I'm going to go over is something that's only just begun to be published. It's a variant of high-resolution melting called high-speed melting. Obviously, it's going to be faster than typical high-resolution melting. And high-resolution melting in general, as it's on real-time instruments today, the melting rates are relatively slow. 0.01 to 0.1 degrees centigrade per second. So that may take half hour, an hour, or possibly even longer to melt. And it turns out that those speeds, the slow speeds, are not necessary at all. The original prototypes were would give melting curves in approximately 2 to 5 minutes. But as it was adopted to other platforms, one of the easier ways to increase your resolution is to just slow down your time. But again, it comes back to instrumentation. If you're focused on high-resolution melting, you can do this at more rapid speeds and get melting curves in minutes. And the interesting thing is that faster, in this case, is better. So I'm going to show you better results with melting curves at 1 to 10 degrees centigrade per second. It's better because it's easier to detect heterozygotes. And I believe this presents a future opportunity for faster and better diagnostics using high-speed melting. So here's one example using that initial high-resolution melting prototype back in 2003 or so. And it's just showing you 2 different rates. The black is a homozygous wild type. The blue is a homozygous variant in SNP. And the red is the heterozygote. And if you look at the faster melting curve on the right, it's obviously easier to distinguish both the homozygotes and particularly the heterozygotes than the slower melting curve. So this is not intuitively obvious. You might think that slower is better because it's more careful, but it's not the case in melting analysis. Faster is better. It makes the diagnosis, it makes the genotyping easier. It makes the identification of sequence variants easier. So here we've gone from 0.1 degrees C to 0.64 degrees C per second. I'm going to show you something even faster done on an instrument from Canon US Life Science, where we're looking at 1 to 10, actually 1 to 16 degrees C per second, showing that the area that the heterozygote is displaced continues to increase as the ramp rate increases. So again, quite powerful example, even at rates of 1 to 10 degrees centigrade per second. So we're talking about melting curves being completed in seconds, which is an interesting proposition in terms of analysis. For instance, you might think about doing very rapid PCR, so-called extreme PCR, followed by this really rapid melting analysis. The PCR can be done in less than 30 seconds. The melting analysis can be done in 5 seconds or less. So it makes an intriguing combination and possibility for future rapid diagnostics. Okay, in summary, DNA melting is simple. The only thing that's happening is those two strands are splitting apart. We're watching that either with dyes or with probes. It can and should be fast, although it's not always implemented quickly on today's real-time PCR instruments. There's lots of information here with little effort, and it's quite inexpensive. So there's our melting curve, just for you to observe at the end, a very simple process. And I have a lot of people to thank for input over the years on making melting better through increases in resolution and speed, including BioFire, Canon, who offers commercially some high-resolution melting assays, both small amplicon and unlabeled probe. ARUP, our reference lab here at the University of Utah. Roche, the original, the licensee of the light cycler that made it a very popular instrument. You also notice on the picture below, the light cycler was featured on an episode of House that we were very, very proud of. And thanks to the State of Utah and University of Utah as well. On the right-hand side, you might remember when we talked about those 3 parameters of temperature, time, and fluorescence. So the projections of the 3-dimensional image are shown, both for temperature time curves, the real-time curve over on the right, and that fluorescence temperature curve for the DNA melting on the top left. I thank you very much, and happy melting. Bye-bye.